Recording in progress.
I'm sorry, give me a second, it's not.
Well,
I think I would know how to manage Zoom by now.
No.
Nobody. Okay.
okay, it's time. Thank you all for coming in today. Today is the last episode of the seminar series for this semester. And we are very thrilled to have Dr. Rachael Seidler here from University of Florida today. She's a professor in departments of applied physiology, kinesiology, and neurology at the University of Florida, as I said. And today we'll hear about microgravity effects on human brain and behavior. Please thank you for coming in.
Yeah, thank you. So I hope everybody can hear me okay on Zoom. I'm happy to be here. I am going to spend the first just couple of minutes giving you a quick overview about UF Space Institute, and then I'll dive right into my research. And I'm comfortable with taking questions during the talk. I told Julie if I feel like you're derailing me too much, I will just let you know, and we will move on. So, UF, we have a relatively new space institute, nothing like the lifespan of yours. We started the Estrella Space Institute a little over a year ago. We received some funding from our university president's strategic funding initiative to set up an institute. And we are not now a brick and mortar institute, but we are renovating some space for a mission operations control center, and we have a couple of offices. So basically, we are faculty just distributed across campus. And this is our leadership team. So Rob Furl is the director. Our position of director is actually currently open. I'm the deputy director. Jordan Callahan is our assistant director of administration, which means she really takes care of all the problems. And then we have assistant directors and a leadership council. And I'm just going to briefly tell you about each of those people and their initiatives, because I think it'll give you a sense of the breadth of our institute, which is extremely broad. Basically, anyone who is doing or is interested in doing space science at UF can become a member of our institute. So our assistant directors, we have John Conklin, who's in mechanical and aerospace engineering. He does a lot of mission payload type projects, designing and launching sensors to detect gravitational waves and other things. He's been really successful at getting funding for these, mission level funding on the order of $20 million and up. And so his role as our assistant director is to offer workshops, training, and support for groups of faculty on campus that are putting together this size of proposal. I don't know about you guys, but UF does not have a ton of experience with the central grants office for proposals that are, $100 million. And so having somebody and a little extra help, grant writers, graphic designers, et cetera, we're trying to get more folks into this level of funding. Jamie Foster is also one of our assistant directors. She is in cell and microbiology, and she has a lab right outside Kennedy Space Center at the Space Life Sciences laboratory building. And she has been there quite a long time. She's very connected with the Cape and with companies in that area. And so she is working with us on corporate engagement, corporate partnerships. Terry Harpold is a professor of English. He studies science fiction literature. So now I think you're getting a sense of the true breadth of our institute, right? So he runs our seminar series. We have a seminar speaker usually every other month during the academic year. He also does a lot of public-facing events. So when authors come to town, like on a book tour or something, he'll have a reading, if it's a space-related book or poetry reading, etc. So he's very good at helping us to communicate with our community, our general public. And then Amy Williams, she is a Mars geologist, and she is helping us with student outreach, academic programs. creating certificates, partnering with our student organization, and so on. And then this leadership council, these are department chairs, center, and institute directors on campus where their work is also space related, so that we can stay in touch with what they're doing and interact and also get their feedback because we're such a new institute. So our goals, our priorities are basically because we are funded as a research institute, our primary goal is to enhance and support faculty research at UF, to facilitate collaborations, to provide leadership, to offer additional support when people are preparing grants, etc. So that's kind of that top circle. On the bottom right, we're hoping to be a go-to resource for our community, the state, of course, the nation as well, with partnerships at the government level, the academic level, the corporate level. And then we also are hoping to inspire with public engagement, with leadership. We do some other things in our community as well, like telescope nights and things like that, programs, outreach with high schools and things like that. So that's a quick overview of our institute. Does anybody have questions or? Julie, I should pick your brain at some point about it.
So how many faculty do you have associated?
Yeah, so we're still accumulating faculty, but right now I think we're around 60. Yeah. How many do you guys have?
Faculty?
Yeah.
No, that's not, 25.
Okay,
Yeah,
okay.
yeah.
Yeah, and because we're so, hey, Helen, because we're so incredibly broad, that allows us, to have
Well,
more
so actually,
members.
we are, let me correct, because, since you're not a brick and mortar one,
Right.
like 25 is
In the
this-
building,
Yes.
okay.
Like, we're currently running the affiliation campaign, so we just got, I don't know, we already had 5 affiliates, we just got 5 more.
Uh-huh.
So, I mean, it's gonna be more than just
Yes.
here.
Okay,
Yes,
okay.
Yes.
And it has to be a faculty member at UCF to join or as an
Well,
affiliate? Yeah,
no.
we get asked that question a lot. Curious. We
that
have
that
faculty-
campaign
faculty members,
specifically is
postdocs,
to
research
consolidate at the
scientists.
UCF level. So
Okay.
this campaign specifically, but we
Yeah.
do have courtesy appointments
Yeah,
with
interesting.
people from elsewhere.
Okay.
So there's a couple of those.
Uh-huh.
Yes.
Okay.
yes.
All right, JD, you want to say something?
Yeah, hi Rachel, this is JD. Sorry I couldn't be there. I had on your slide when you talk about the priorities, I don't see engineering, I only see science.
Yes. So, right. Engineering, of course, is applications as well as science. And because JD, our funding came from University President's Strategic Fund, so it's through the Office of Research. And so engineering research absolutely is part of this. In fact, a lot of these categories that are listed on that slide are actually part of our engineering. A large number of our members are faculty in mechanical and aerospace, as well as material science engineering, electrical and computer engineering as well. So these are sort of more topics, I think, rather than departments. Yeah.
Okay,
I mean,
thanks.
like any
Yeah.
one of those needs engineering to fly the system.
Absolutely,
So, yeah.
absolutely, yes.
All right, Anis,
Yeah, I am part of this seminar series which I think the physics department runs. I was wondering whether your seminar series is also have an online component so that we can also connect from here.
Yes, for some of them we do, but not all of them. It kind of depends on the speaker and where we are. We try to make those seminars sort of a faculty and student gathering touch point. So we don't always offer them on Zoom, but if you go to our website, which is astreas.ufl.edu, you can see our calendar of events. And if there's one in particular, for example, if it's more physics focused, I would say just reach out. We'll see what we can do.
Okay, thanks.
Sure. Okay, any other questions about the Institute before I switch gears and talk about my research?
Yeah,
go
I mean, I don't know, it's maybe a very technical question.
Sure.
So you get some internal incentive for a
Yeah.
certain period of time?
Yes.
Okay, how much
Yes.
is that? 5 years? I don't know.
Yeah, it's for 5 years. It sounds like a lot of money, but it's actually not. $2.5 million.
Oh, I know.
Yeah, you know.
Nothing is ever a lot of money.
We have a standing operating budget from the Office of the VP for Research, which is somewhere around 500,000 a year, but they would, of course, absolutely like us to move off of that. They also are supporting the renovation of the space for our Mission Operations Center.
But like all your affiliated faculty, they're all, tenure track, tenured kind of people.
Yes, also research scientists. Some of our
Uh-huh.
members are postdocs.
Okay.
Yes,
So you get
Yes.
some soft money people?
we do.
Okay.
We do. And again, being a new institute, it's definitely slow going in terms of like development campaign type activities. We do ask for, I don't know if UCF has the same, UF has a certain portion of faculty indirect costs can be allocated to indirect cost return allocated to centers and institutes. But that also does not provide a ton of money because many of our faculty are members of, like I'm a member of one center and 2 institutes, so that splits my money.
Yeah.
Yeah.
Okay.
Yeah.
All right.
Yeah.
Yeah. Good.
Yeah, thanks. Okay.
Congrats.
All right, then I will go ahead and change gears and start talking about research. So I have been studying human brain and behavior changes with spaceflight and analog environments since I was a graduate student. We won't say how many years ago that was, but it was quite a few. And I started in a lab where actually I was working on aging and studying sensory conflict. So what happens as we age and our sensory systems degrade or get noisier, and they're not sharing the same information. And I realized that, this also happens in spaceflight. I'll talk a little more about that. And then I realized, NASA has graduate student fellowships. And I thought that would be a cool thing to apply for. So I reached out to someone who was the director of the neuroscience laboratories at Johnson Space Center and naively said, here's my idea for my fellowship application. And this person was very kind and basically said, no, that will not work, but let's schedule a call. And ended up kind of helping me to shape the project. I got the fellowship. I've been collaborating with people there ever since. This person was longtime friend, collaborator, mentor. He just retired a couple years ago. So I know, I think a couple of you said you're students, just encouraging you, don't be afraid to, to reach out and contact people. So when we first started thinking about how the human brain and behavior and human performance might change with spaceflight, I really thought we would see extensive neuroplasticity, right? So imagine our physiology has evolved with gravity. A lot of our physiological systems work against gravity. Our muscles contract and it sends fluid and blood up. The heart pumps up. We have lots of specialized or one-way valves. Some of our physiology has evolved to actually work with or leverage gravity, most notably the vestibular system. When you tilt your head, gravity pulls calcium carbonate crystals across the hair cell receptors. So I thought, wow, we're going to take people and they are outside of an environment in which they evolved and humans, animals evolved. And not only that, but when I'm studying this kind of thing in my lab, I can get people to come into the lab, maybe participate in an experiment, practice a motor skill or a cognitive task, maybe for an hour. Maybe they'll come a few sessions if it's an intervention or training study over some weeks or months. But here, people are exposed to this microgravity environment 24 hours a day while they're there, even while they're sleeping, right? So what an incredible adaptive stimulus. And I will tell you that we found some evidence for neuroplasticity, but also a whole bunch of other stuff we did not expect. So I'm going to share some examples of both of these today. And again, by the time we started this study, we started doing research, brain imaging of astronauts around 2010. We already knew a lot about how human behavior changed. We know when astronauts come back, they are not just carried on a litter because we so highly revere them, but because they're incredibly dizzy. They have poor balance. They have orthostatic intolerance. They might faint. They might trip or fall, right? So we know there are lots of sensory motor after effects for this in-flight adaptation. There had also been lots of experiments with non-human animals flown in space, showing changes in synaptic connections in the cerebellum, changes in the corticospinal tract connecting the brain and the muscles, changes in neuromyelination, etc. There were also some really cool experiments done with this toadfish model, which actually has a vestibular system. They've been flown in space many times and shown some really interesting effects when they're in a microgravity environment. These calcium carbonate crystals in their vestibular system, the system responds by making them bigger, as if the nervous system thinks that would help them to detect gravitational cues in the absence of gravity. If you take these animals and spin them in a centrifuge, those stones become smaller. So there's really interesting animal adaptive responses.
Does it work? When the crystals are bigger?
No,
Yeah.
no,
Okay.
because there's still no gravity, right?
Okay.
To pull them, still no
If
gravity.
it's low gravity, would it work?
I think it would work if it's low gravity.
Mhm.
Yes,
So
yes.
it's kind of a good response.
It's a really cool response. Yeah.
Okay.
Yeah.
It's weird that it should be that smart.
I know, right? Yeah. So, okay, a little bit of audience participation for you guys. I know many of you are space scientists. Which of these people is right side up? Okay, you're laughing, which means you know the answer, which is none of them, right? So, when people go to the International Space Station or when they're in microgravity, particularly the otoliths portion of our vestibular system, this is the part that is sensitive to linear accelerations and to head tilt. You still have linear and angular accelerations while you're in microgravity, but this head tilt business, so if you want to all just quickly try it with me, you can take your right ear and tilt it towards your right shoulder. So you can feel it in your neck muscle sensors. You can see it visually. You can stop doing that. But on Earth as
Like,
well.
yes, you live.
So when you do that, again, these calcium carbonate crystals are in kind of a gelatinous goo in the otolith. And when you tilt your head, they slide across these hair cell receptors, which is, I think this is another really cool fact about these. They are biomechanically, not electrochemically gated channels. And so they biomechanically get pulled open by the stones. So in microgravity, that does not happen. And, when you're in an environment like this, the crew often report severe space motion sickness when they first go up. It's because of this sensory conflict. So your neck muscle receptors and your vision would still tell you, yes, your head is tilted, and your vestibular system would say, no, nothing happened here. And that sensory conflict is really provocative, makes people feel sick. They also have really interesting kinds of illusions where this person might feel like they're upright, and then in the next second, they might feel like they're upside down, and then upright, and then upside down. So it's very disorienting. The ISS is also a really visually disorienting place. There's no cues of floor and ceiling because there's equipment everywhere, right? They have to make maximal use of the space. So what looks like, a wall in a lab to us that's full of racks and computers and so on, the floor will look like that too, and so will the ceiling. So there's not really visual cues to help either. And our somatosensory system, our sensors in our skin, our proprioceptors are also unloaded. So these are sensors in the bottom of the foot. So you're no longer standing on them, right? You can locomote from one end of the room to the other just by tapping your finger. You're not contracting like large anti-gravity muscles in the legs all day because you're not standing against the effects of gravity. So these sensory systems are also altered. They're getting greatly reduced inputs. So this is a video of a crew member that he publicly posted. So it's in the public domain. And this is shortly after landing. He is trying to walk a straight line with his eyes closed. And you can see he's really struggling, and it's not because of loss of strength, right? It's because of difficulties with sensory processing, sensory integration.
It's not the Russian vodka either, right?
And right, exactly. I mean, maybe. And this has been quantified as well. So this person is standing on a particular balance measurement that they use quite often at Johnson Space Center. It's a neurocom equitest. So the person is standing in it, their eyes are closed, they're standing on a sway reference plate, which means as they lean forward, the plate also tilts forward. So they're not getting this like change in ankle joint information from their receptors. They also ask the crew members to nod their heads while they're doing this, and it's incredibly difficult. So here are pre-flight scores, and blue and green here are just different duration flyers. Pre-flight, a perfect score would be 100. So even pre-flight, people are kind of struggling with this. But these are the days post-flight between day 1 out to day 10. And you can see some people are fairly well recovered within about a week. Others are not, even at 10 days. There's one person who's a rock star when they come back. There's obviously huge individual differences, which is a very interesting question in
They're
itself.
doing better than
And
what before they
I
left.
know, right? And something that I work on in my lab is it's trying to predict these individual differences, but also trying to understand what is changing in the brain when this is happening. And the behavioral literature, when we started, the behavioral evidence suggests that the way people deal with these confusing sensory inputs in flight is that they shift their sensory reliance, right? So the brain starts to learn that, hey, this otoliths portion of the vestibular system is garbage. It's not telling me anything useful. So I'm going to start ignoring it. I'm going to stop listening to it. And when people come back, there's an after effect of this, right? So you come back and now you're in Earth gravity, and it's like your brain is just being bombarded with these cues that you have not used, paid attention to, or even had available for months or a year. And so this readaptation takes time as well. You cannot just be immediately back in context. Yes.
So it seems that these two groups were kind of selected differently. I don't know, like
They seem a little bit different at the beginning.
yeah,
and
the green people
the green and the blue, yeah.
were
So
doing
this,
better.
Yeah, so this paper is about 10 years old. This is kind of a little segue here, but I think it's interesting and related to your question. For a while, NASA thought that people who had flown more than once adapted better. And there's maybe some anecdotal evidence for that. And usually the people who have flown more than once are long duration, then they become long duration flyers, or they are long duration flyers. You've proven yourself, you're highly skilled, but they know you can work in this situation. So these two things seem to be intertangled in the data sets, whether you've gone before and whether you're a long or short duration flyer. And so that seems to be part of this maybe pre-flight effect, right? But since this time, they have, this measure is used on almost all astronauts and has been for quite a long time. So in the last 10 years, they've done a little more digging in and said, at least with this measure, those things don't seem to make a difference,
Mhm.
but open question.
And
Yeah.
overall, green is better afterwards.
Yeah.
Yeah.
Yeah, right? Yeah.
Have you ever sliced this in terms of, traditionally, right, NASA, selected a bunch of military aviation people.
Yes.
Navy guys that spend a lot of time on aircraft carriers, bopping
Yes.
around.
Yes.
And have you sliced this data in terms of those people that they're used to their vestibular systems being
Right.
totally hosed up?
Right. So we have not. We have looked at some other factors, some of the obvious ones like age, gender. Have they been before? How many days in space have they spent previously? We have not done what you are asking. It's interesting. I mean, potentially we could, but I think you would be left with, it's the same thing in comparing men and women.
Yeah.
So few women have gone relative to the number of men, so few people who came through a civilian route rather than a military route as well. But good question, and I think that's changing, actually.
Don't you, it seems to me that, I've flown on experimental aircraft.
Yeah,
And
yeah.
there's some people that just can't handle it.
Yeah,
And then
absolutely.
there's other people, I personally have never been airsick or seasick, and I've spent a
Okay.
lot of time on
Okay.
airplanes and on boats.
Yeah,
And so,
so, yeah,
What is-
so,
Criteria is
So-
that it's not really related.
so I don't know that that's part of selection, but there definitely have been studies looking at sort of related to that, not necessarily motion sickness, but like, can we look at how adaptable your sensory system is to sensory conflict, and might that help you? And the answer seems to be sort of.
what I was wondering in terms
Yeah.
of what you do
Yeah.
is that an opportunity to look for, genetic markers and differences in
Okay.
those people that allow them to take up that vocation with, and if
Yeah.
they fit in great, and maybe they're good at this too.
Yeah. So there is an ongoing study called Predictors, where I'm not the PI, but I'm a co-I, and they are looking at some genetic markers.
Okay.
They're looking at dopaminergic genotypes, so genes that code for different aspects of the dopaminergic neurotransmission. There's another one, I forget what it's called, but it has been potentially linked to vestibular function, but a little more the cardiovascular regulation side of it than the orientation side. And there are some preliminary results that suggest that maybe these things carry a little bit of predictive power, but not as much as some other measures, which are different measures of like balance or adaptability before you go. We also have a paper under review looking at a particular measure of how the brain processes vestibular signals that seems to also predict some of these individual differences.
Interesting.
Yeah, it is
Are
interesting,
we
right?
derailed by now, or can I keep?
Yeah, exactly. I mean, yeah, go ahead.
So I heard the legend
Yeah,
that women overall do better, and it's not just space flight, but also in those parabolic flights, that they
Yeah.
just proportionally they end up less,
yeah,
puking in the bag.
yeah.
Is that true, or is it not enough women, like you said?
So I don't know that much about the puking in the back thing, but I do know a lot of the literature on space physiology, now that there's more women available in the data sets. Some of these studies are, it's like 25% women and 75% men, so it's hard to say. But what I can tell you is that women are not, what's the right way to describe it? Not more susceptible to sensory motor declines, cognitive declines, brain changes that we would call dysfunctional. In some cases, some of the studies on sort of demographics of spaceflight associated neuroocular syndrome suggest women may actually be protected from that for some reason. So all of that to say, women are at least equal in their responsiveness. I don't know about the puking in the back.
But you know, like I mean, that would be another question. Like, there's these parabolic, sorry,
Yes,
aircraft.
yeah.
So is that a better indicator compared to like whatever?
I don't know. I do know a lot of people do sort of pre-training on those. I actually have a project this summer with Space Force cadets on parabolic flights.
Are you going?
I hope I can, but I'm not sure if I can. I might just be putting sensors on them, so we'll see. But yeah, I again, it's also a little bit different, right? Because it's 20 to 30 seconds of microgravity and then hypergravity and then normal G, and then you do it over and over. It's probably more provocative, actually. But I don't know if it's a good predictor
All right,
or
I'm
not.
just a bit.
No, these are great questions. Yeah. Okay, ready to take a step further though. Okay.
Mhm.
So this big study that I will tell you about, we basically wanted to collect all kinds of behavioral measures. This is a photo that Kate Rubins posted of herself doing one of our cognitive tests on station. But pre and post flight mobility and cognition, some cognition in flight, obviously they can't do the mobility measures when they're floating, and pre and post flight measures of brain structure and function. A lot of our behavioral measures were ones we already know change. And the question was, how do they map onto brain changes? Are they associated or are they not? And this was our experimental timeline. So we tested people twice before they went to station. While they were on station, they did our experiments 3 times, unless they were there for a year. Then they did our experiments 6 times. We capture them around day 4 when they get back, and we follow them out to 6 months post-flight. So for any given participant, this is at least a 1.5 year timeline. So how long do you think it took us to collect the data? We ended up with 13 participants. Yeah. How long do you think?
10 years.
10 years, almost exactly 10 years.
Yeah.
And this was during the pandemic, and we had collaborators on the grant that were collecting the data at Johnson Space Center who put themselves in quarantine so they could collect our data. So we tried to keep them happy. We made them blue moon cookies, moon landing cookies, all kinds of stuff with my lab, and sent to them, and sent, all kinds of thank you. But by the time we collected all of the data, it was roughly 10 years. So you better hope, if you're starting one of these flight studies, whatever question you're asking in the beginning, you hope is still relevant a decade later. Yes.
So that's interesting, right? So that was during classical space flight setup where you would have federally funded astronauts. So
Yes.
there's
Yes.
like this boom of people
Yes.
going.
Yes.
How's that going? Probably going your way, right?
So, I mean, this is, I'll give you the short answer. We can talk more about it later if you want, but yes, we are involved with, I am doing a couple of studies with a couple of different commercial crew companies. But here you were talking about federal funding. I feel like the way it's set up right now is terrible for scientists, because the companies are saying our travelers want to do something meaningful in space. So scientists are basically providing their in-flight entertainment. Scientists are not getting any money. They are being asked to travel themselves, set everything up, provide their own equipment, et cetera, et cetera. So the model right now is not great.
But you're getting free
But
data.
we are getting access.
Yes.
Yeah.
I know.
So it's very
Yeah.
interesting.
Yeah.
But I feel like it generates a lot of inequality, right? Like people at small schools, junior faculty,
yeah,
people who don't have like a slush fund budget cannot do
This
this.
may be just a matter of educating these industry people. They might not know if
Yeah.
you're not on a salary, you know.
Oh, no, they know. We are trying.
Oh.
Yeah. Anyway,
Yeah.
okay. So when we
Like
started,
She said, yeah, just don't get it.
yeah. It's not in their business plan.
Yeah.
Anyway, when we started, we hypothesized that we would see extensive neuroplasticity, particularly in this part of the brain. It's called parietal operculum 2. It receives a lot of vestibular projections. It also receives inputs from your visual system, your somatosensory system, but it's the main cortical vestibular input area. And as I touched on briefly, we also saw what I will describe as evidence for direct mechanical effects. So I'll tell you about some of both of these. And the latter was a little bit surprising because it is the overwhelmingly dominant change that we saw. The neuroplasticity is sort of hidden underneath. Yeah.
For those of us who don't know, what does neuroplasticity mean?
Okay, so structural or functional change in the brain in response to experience. So like if you're learning to play the piano, you might see some changes in sensory and motor and auditory regions in your brain. And we would call that neuroplasticity. And it's thought to be a relatively permanent change.
Oh, okay.
Yeah.
And mechanical effects, what are those?
So mechanical, absence of gravity allowing fluids to shift upward
I see.
and
So
fluid
actual-
shifts impacting the brain.
Because of
Uh-huh.
there's no gravity.
Yeah, yeah,
load on it.
Exactly. Exactly.
So actual
Actual,
matter moving around.
yes,
Okay.
and pressure and.
Yes.
Yeah. Okay, so, but let's first talk about the sort of neuroplastic or structural and functional changes that we think reflect or relate to the process of adapting. So first of all, some of the MRI measures we use, we can look at gray matter, some of them we can look at white matter. So this is a particular analysis of white matter changes. And in particular, the white matter changes that we see are in the cerebellum, which people tend to think of as being like a coordination part of the brain. It receives all kinds of sensory inputs. It's involved in control of movement. We also see a lot of changes. All this stuff here on the side is the white matter underlying this vestibular processing region. We also see a lot of white matter changes in the corticospinal tract, which connects motor controlling areas in the brain with the motor neurons in the spinal cord, which then interface with the muscles controlling movement. And all of these changes are in the direction that you would expect to see with aging. And we look at there are huge publicly available databases of this kind of scan. So we analyze those data as well and look at what would be the rate of change of white matter in the brain of someone approximately the same age and sex as the crew member and over the same period of time. And these are 3 to 5 times greater than what you would expect with aging during this time frame.
But does this mean that they age faster or?
Not
it's the opposite?
necessarily, no. I it seems to be more like these changes, even though I'm saying neuroplasticity, which sounds like a positive thing, these are probably more sort of white matter, if you want to describe it this way, atrophy. So you're not getting the same amount of vestibular inputs, you're getting degradation of white matter in that area. You're getting degradation of white matter in these pathways that are sending motor commands down and bringing sensory signals up, because your movement is very different, right? You're not contracting your muscles, you're not getting somatosensory inputs on the bottom of your feet, etc. So there is some recovery over the 6 months post-flight. So that's why I would not say it's aging faster per se. It's flight related. Also, the magnitude of the effect correlates with how long you were in flight. So it's definitely a function of the flight.
So you're saying it's kind of analogous to what you see in aging
Yes,
individuals?
yes, it's in the same direction as what you would see with aging. Yes.
But it goes back whenever they.
It doesn't go all the way back. So I'll show you a few examples of that as well. Okay, so we also find that the larger the white matter changes in this region underlying the vestibular cortex, the worse your balance is when you come back. So if you think about that graph I showed you with a huge spread of changes in balance, people who show the biggest decline in balance from pre to post flight have the largest changes in this white matter region. So we also, leading up to this study, we developed a way to stimulate the vestibular system while people are in the MRI scanner. There are lots of ways you can do this. Not all of them are pleasant. So one involves putting incredibly cold water into the inner ear. We did not want to do that to the crew members.
Gives me a headache thinking about it.
I know. There's another one that involves a
This sounds like.
very, yeah, exactly. A very, also very high decibel tone burst.
Oh.
I tried that one in the scanner and I kept saying to my team outside of the scanner, I was like, who is moving the screen? And they're like, the screen's not moving. So every time it went off, I had a vestibular reflex response that my eye muscles would contract. And so I thought the screen was moving, but it was just my eyes. But it was so loud that I came out and I had like a headache on the side of my face where the sound was delivered. So we said, we need to do something else. So we took an existing clinical test and made it work in the scanner. So this involves, it's like using one of those little tendon tap reflex hammers, but you tap either the forehead or the cheekbones. And it's not that it moves the head, but actually it starts the bones vibrating. And through bone conduction, the signal gets transmitted into the inner ear, and you get a vestibular evoked myogenic potential in the eye muscles. So if you put little recording electrodes under the eye, you give the tap, you have a characteristic waveform that starts roughly 20 milliseconds after you tap. You get an eye muscle contraction. You can also record this in neck muscles. So we worked with a company that makes pneumatic tappers, and we set up a compressed air system with really long tubing to get it into the scanner from the compressed air system outside of the scanner. But we actually were able to use this to do a bunch of studies on age differences and vestibular responses and how those relate to balance in older adults. But we also did this in our crew members. So we had done this now in many studies with control participants, and our crew members pre-flight look very much like the control participants, where these areas in orange, again, this is that vestibular processing brain region, are active. This is also a region in the cerebellum, our sort of coordination structure. And then these areas that are in blue show deactivation or suppression. And this is very typical when you're looking at the brain in terms of sensory stimulation. Whatever sensory channel you are stimulating, you get an increase in activity in that brain region and suppression of activity in other sensory regions. So this is extensive suppression of visual cortical areas, somatosensory and motor areas while we're delivering the vestibular stimuli. And if you look at these other sensory areas, those are the ones that changed their response to vestibular input the most from pre to post flight. So this is what it looks like post flight, where we are delivering vestibular stimulation. And now this vestibular region is not changing. What's changing is you're recruiting more of the visual areas, more of the somatosensory areas. And if we look at that kind of averaged over time, here's our two pre-flight time points, right? So 180 and 60 days before people go. This is 0. So these areas, when we deliver vestibular stimulation pre-flight, are suppressed. People go to space and come back, these areas are engaged. So this is what I was referring to as sensory reweighting or sensory compensation. The system is basically saying, I don't know what to do with vestibular cortex right now. I'm going to recruit some other areas to help me out. And they're still elevated above their baseline, about a month later, but things kind of go back, 3 to 6 months later. And interestingly, this is seen in studies where people have been blindfolded for 5 days. And during those 5 days, they were teaching them to read Braille using their fingertip. So before they were blindfolded, they get stimuli on the fingertip, activates this somatosensory processing region of the brain. 5 days, no visual inputs, a lot of tactile inputs. You put them in the scanner and tactile stimulation then activates the visual cortex. So it's like the sensory systems are saying, I'm not being used. I'm going to give up my territory for something else. So, yeah.
So when you first started this whole line of thing with the tapping sensors and all that, I was gonna ask you a question if anybody had thought of putting haptic sensors on like your soles of your feet.
Yes.
And maybe tying them to the inertial system in the space station. So
Yeah.
give them an actual reference.
Okay. So a Russian group has done this study.
Okay.
Where they call them gravity boots. So they deliver stimulation to the bottom of the foot, kind of following the pattern of how people step. So, you press on your heel and then push off on your toes. So it buzzes the heel and then under the toes. And they did functional MRI of this pre and post flight. And they found not necessarily changes in activation, but changes in connectivity of different brain areas. And they, unfortunately, I don't think they had a big enough control group that did all the same pre and post flight stuff. They also did not look at does this stimulation preserve balance?
Yeah.
But it seemed to result in a different pattern of functional connectivity between sensory areas post-flight than people who did not have it in a way that might suggest you're preserving some of this.
In the hopes of using those same sensors. Maybe they're not the same, they don't get the same exact thing back, but it's the same area of the brain involved.
It's like a similar stimulus,
Yeah.
right?
Yeah.
Yeah. So I think it's a really cool idea. Yeah. Okay. The problem though is the vestibular input. I mean, you can stimulate the vestibular nerve with an electrode on the back of the neck, but that's sort of like hitting all vestibular inputs at once. It's not just getting the part that's affected.
Yeah.
So
There's no clean way to do that.
no, unfortunately. But another thing we see is that people who do this cross-modal sensory recruitment the most, these folks actually have the best balance post-flight, which suggests to me that this is a compensatory kind of neuroplasticity response. So if we look at this cluster in the somatosensory cortex, here's the difference score in balance. So these people, their balance gets worse. Here's one person whose balance actually gets a lot better. So people whose balance is getting better or declining the least are recruiting these somatosensory and the visual areas more post-flight. Again, suggesting that this sort of sensory substitution thing is a compensation response. We see the same thing in visual processing regions. So same direction of the relationship where people who recruit visual areas more in response to vestibular stimulation have better post-flight balance. Okay, so I think that this provides pretty good support for sensory reweighting. So again, imagine microgravity, you're getting altered vestibular signals. You respond by kind of suppressing vestibular signaling and compensating by trying to upweight more of the vision, maybe the somatosensory as well. And then post-flight, we see some lingering of this. So what we call after effects that again will last for weeks to months while people are relearning how to move in the presence of gravity. So for the first part of the talk here, I've kind of explained brain structural and functional changes that relate to sensory motor function changes with spaceflight, right? And there is some evidence, this is the kind of stuff that we were expecting, but I'm going to change gears a little bit and talk more about the stuff that we were not expecting. And in hindsight, of course, hindsight is 2020. We should have expected some of this, right? So, of course, we already knew there are fluid shifts when people go into microgravity, right? There are so many photos of astronauts when they first go to space and their faces are puffy and, their legs are super skinny. And there's some, while you're in microgravity, there's some sort of equilibration, but people actually lose up to 1 liter per leg of fluid. Okay, that's a lot while they are in microgravity. And then when you come back, things come back down. This is also what contributes to orthostatic intolerance, right? Not enough fluids up here, you stand up quickly and you faint. So we didn't really think about how this might interact with stuff in the intracranial compartment inside the skull, right? But I'm going to show you some changes in brain position shifts and changes in fluid distribution in the brain that are actually visible by eye. So this is looking at the brain on the side. This would be where the eyes are. This is the back of the brain. And I've got some red arrows here, meaning please look here. So pre-flight, you can barely see this black line between the brain and between the dura. And this black is cerebrospinal fluid. So I'm going to toggle back and forth between pre-flight and post-flight. So the skull is aligned, right? But you can see the brain is physically shifted up and that cerebrospinal fluid is pushed out of the way. The brain is actually kind of compressed up against the dura, okay? Another part of this that is visible by eye is looking at the ventricles. So these are the large fluid-filled spaces in the middle of the brain. They're full of cerebrospinal fluid. What we've done is we've used an automated algorithm pre-flight to draw this red line around the 3-dimensional boundaries of the ventricles. And here, when I toggle, what I want you to look for is you'll see the black extending beyond the red dashed line. Where's my mouse? There. Okay, so you can see it around here quite dramatically along the top here. So in some individuals, the ventricles expand up to 25% of their volume. Okay, so you can imagine this is the ventricles in the middle are expanding out. They're compressing white matter around them. The brain is shifting up. These structures at the top of the brain are being compressed. This actually, other people in the field see the same thing with European, Russian, etc. Crew members. And it's been a really interesting challenge for the field because our typical data analysis techniques don't even work. The gray matter is being pushed against the dura, which is also gray. It's so pushed together that automated algorithms can't draw a boundary and separate them, which makes it hard for us to say, did the gray matter volume change, which would be a neuroplastic response? So there's a lot of questions. One question relates to how much does this happen? Does it depend on flight duration? So this is a paper we published a couple years ago showing this. Each of these panels is just a different ventricle. So one is on the left, one is on the right side of the brain, one is in the middle. And the first data point here is 8 people who went to space for a few weeks. So you can see the people who went for a few weeks, basically no change in left or right ventricle, but third ventricle volume is definitely bigger. Here are 18 people who went to space for about 6 months. So here, here, and here. And you can see their ventricles expanded quite a bit. Then we have 4 people who went to space for a year. And this is the point where we kind of went, oh, phew, it's not a, continuing linear function. It's maybe still going up and it's only an N of 4, so we don't really know. But there's some sort of flight duration effect. And then some of these folks in this data set, which was 26 people, 28 people, were repeat flyers. So we were also able to look at their pre-flight for this flight, ventricular volume, which seemed to be still expanded if they had flown within the last 3 years. Okay.
Oh,
Right. So this suggests this needs at least 3 years to recover. Now, recover, what does that mean? Well, these folks are not complaining of headaches. If a neurologist saw this on Earth, they would say, we need to get you to the hospital. We need to put in a stent. We got to do something. No headaches. Very little, if any, cognitive decline occurs with spaceflight. And so the question is this really a risk? And I think it depends on who you ask. One of the projects that I'm involved with right now is something called the NASA Long-Term Health Study. There's 5 PIs. One of the things that we're looking at is this effect. So we're following people out to 5 years post-flight. We also have an arm where we are calling back retired astronauts. And the reason I think this is an interesting question is that the ventricles expand with age. It's not that you're getting more fluid pushed up. There's actually structural atrophy around the ventricles and the space just gets filled with cerebrospinal fluid. So it's a very different mechanism. But I think the question is, how does this interact with the aging process? Are these people more susceptible to neurodegenerative disorders, cognitive issues, etc.? And you were going to ask something, or?
But I think you were answering.
Okay.
It was more, my question was, what's the problem with the vestibular being?
Yeah,
higher?
yes, yeah. So, yeah, I could talk about that a long time, but I will also show you.
Okay.
This is fluid within the brain tissue itself. And we see kind of similar effects where everywhere where it's blue, there's reduced fluid in the brain tissue. Where it's red, there's increased fluid. And it's hard to get a handle on what's happening here because there's a pretty good covariation with the number of previous missions people have flown in this cluster. But the sign reverses at some point, whether it's a decrease or an increase. So this effect is also kind of hard to understand. It's not that there's overall increased fluid. There's not an overall increase in intracranial pressure. There seems to be an outflow problem, CSF outflow and recycling out, which we, the field, thinks is potentially because of the brain getting pushed up against the dura. There's some vessels that exit the brain up there that are involved in brain waste removal, so lymphatic system, and also fluid recycling.
Isn't some of that fluid, I mean, just an adaptation to protect your brain from injury if you get jarred or?
Not necessarily, right? Because your brain, so on Earth, yes, cerebrospinal fluid, you want it to surround your brain and protect it.
Well, I guess that's what I mean is if
Yeah.
if it's less of it there,
Yeah.
that would seems like you would be more
Could be a
predisposed if
problem.
you got in a car accident.
Around the
right?
top, yeah,
You might
especially
have-
if your impact was
Right.
up. Yeah, I think so. Yeah. Yeah.
So I think what Esther was asking is like, because you said if some patient was coming in, an Earth
Yeah.
patient, and had these
Yeah.
extra fluids there, you would send them straight away to the hospital.
Yeah.
Would they have headaches?
Headaches, for sure, they would have headaches,
So why is this? people not
potentially
having
blurry
headaches?
vision. That is also a really good question. And another question that I initially was thinking about is why does it take so long to recover? I think it takes long to develop, right? You saw the difference between the people who spent a few weeks and the people who spent 6 months. Now, if you think about recovery when it comes back, it's probably, if you think about it, it's probably not good if it would immediately recover, right? Because you would go and your brain would plunk down, and that might be like a
Yeah.
TBI.
Yeah.
So there's probably some adaptive effect that it takes a while to recover. But also, the mechanisms of this on Earth are different than they are in flight, right? So I think that's partly why they're not like experiencing headaches, dizzy, blurry vision. But also, these people are much younger than what you would see this in on Earth. So on Earth, this kind of stuff tends to happen when people are quite a bit older. They're highly trained, highly selected, very fit. And I mean, this is my personal opinion, but I think the central nervous system reserve is in some way compensating. And I'm more concerned about what's going to happen to these folks 30 years down the line. And you might say, well, what about existing older astronauts? And what I would say is that we don't have a large enough older adult astronaut population yet that has actually flown
Longer.
for long duration. Yeah, we're getting there, but probably like another 10 years, we'll have a better
But it
sense.
will certainly not go the right way for them.
I mean, listen, I will say, I don't think that it will, but at the same time, so many people know this. They don't care. They would go. They're sitting on how many tons of rocket fuel
Oh, yeah, no, I'm
that
not
are being
saying
set
that.
on
It's
fire.
the thing,
They
yeah.
don't care,
I'm
right?
worried about the extra radiation.
Right? And so, some of the people I work with feel very strongly that this is a huge ethical concern and no one should be spending a year in space. I kind of feel like, wow, this is really interesting. It's provocative. It raises a lot of concerning questions, but why would we stop anybody? Okay, so some
So I'm
questions.
just a little, sorry. I don't know if I follow
Yeah.
this right away. So a person who is selected to go to space, spend like 6 months or more, they will get this inflammation in the brain in general, or in the ventricular.
Yeah.
And this happen in Earth with aging. That's what you said.
So normal aging, there's a little bit of an increase in ventricular volume, not this much.
Okay.
Very different mechanism.
Okay.
Yeah.
But the reasons are different, I guess.
Absolutely different. Yes.
Okay.
Yes. And then there's a patient population that might look like this. And the mechanisms are very different as well. Intracranial hypertension, normal pressure, hydrocephalus.
So it's related to different in pressure, not
Yes.
only gravity. Like it's not really
Yes.
gravity, it's a
Yes,
different in
and
pressure.
the thing that's been interesting, there have been measures of intracranial pressure in space, but only non-invasive measures. So we don't know how good they are. They suggest that the average level of intracranial pressure is no different than it is on Earth. But what you miss is that on Earth, intracranial pressure changes every time you lie down, sit up, stand, and in microgravity, it's constant.
So is there any different, I guess the difference would be, I mean, I understand the difference in pressure is not as much as when you're in microgravity, but what about people who lives in different at different altitudes that
Sure.
experience different pressure in the brain along their lifetimes?
I think it's not going to be the same magnitude
Yeah, sure,
effect.
but
And again, I think, we have mechanisms that adapt to that. I mean, obviously, to an extent, right? People can get, acute altitude sickness and other much more dangerous chronic conditions like accumulation of fluids and, you know, if the altitude is too high. But yeah. Yeah.
I guess my question is, so whenever they first go into split space, do they like start complaining about headaches like people who first accumulate them from old age do?
So, so with aging, the ventricular increases is not enough with typical aging that we would expect somebody to have a headache. And so they're not complaining of headaches for that. It's more the clinical conditions
Yeah.
on Earth. In space, there are some crew member complaints of headache, but they actually correspond to times when CO2 levels are peaking. So I think it's more of a CO2 effect than a fluid shift. They're not complaining of it, you know, once this happens, it's sort of stuck like that for quite a while. And it's not like they're saying, oh, I've had a migraine for weeks or nothing like that.
Okay.
Which is interesting, right? Which
Mhm.
again raises the question of, do we care? Yeah.
Okay, so now kind of to roll it all up, I'm wondering, can this research inform, like, what would be the lowest amount of microgravity that you'd have to provide in an orbiting station? But you could rotate it.
Oh, yeah.
That would be easy to do, but maybe it doesn't need to be one.
Oh, the
G, it
lowest
could be-
amount of
it could be a
artificial
tenth of a
gravity.
G or
Yeah.
something like that.
Yeah. So, first of all, I have a couple of review papers on here and like public science type articles that I've written if anybody is interested. But what's next? Yeah. And I'm gonna skip down to, we have done a study at the Envy Hub in Cologne where people were on bed rest. And one group went to their centrifuge and was spun every day with a G-force along the long axis of the body. It was roughly 1 GE at the level of the heart, but that means it's like 0.3 G at the level of the vestibular system and where all this
Right.
fluid accumulation is. So people that are being spun had less balance declines than the control group, but it had 30 minutes a day, had zero impact on the fluid shifts, brain position shifts, which are replicated, by the way, in head down tilt bed rest. Not to the same extent as in microgravity, but you see enough of this. 30 minutes a day at that amplitude was not enough to budge anything.
Right.
So that makes me think it needs to be longer
Right.
or higher,
Yeah, I mean, it seems like you could do maybe an animal study or something where you could,
right?
where you could put an animal in a centrifuge and spin them at different rates in
Yeah.
zero G.
So there are also a ton of dose response studies that have been conducted and are ongoing for lower body negative pressure as well, which is, you know, like vacuum pants,
Right.
basically. Yeah.
Interesting.
And there's not hard data on this, but the cosmonauts, you know, they have this Chibis lower body negative pressure that's been on station a long time. They all use it. And they claim none of them get spaceflight associated neuroocular syndrome, but this is totally anecdotal. So, yeah.
Interesting.
Yeah.
So,
Yeah.
Are there any other like interventions that could be done? I guess spin your space station or vacuum pants, I guess, but anything else?
Kind of my question as well, a device of some sort?
Yeah,
Yeah.
so right, I mean, lower body negative pressure is a device. I also have a colleague at Texas A&M. She's working on sort of a smart suit compression garment, which again, I don't know if that would help or not, but it's basically like a suit, and it's not just for spaceflight, but it's like for people that maybe have edema or whatever. So you put the suit on and it's loaded with sensors and it can sense where there is swelling, and where there is swelling, it provides compression. So you could imagine maybe something like that would help, but yeah, I don't know how you like squeeze it down. But at least if you're getting rid of it, you know, in this area, then maybe it's not getting up there, but I don't know. The other thing that's kind of crazy about this is around the same range of time, we've learned a lot about the blood-brain barrier is way more permeable than we thought, right? So you might think this shouldn't happen because of the blood-brain barrier and separation of CSF and the rest of these fluid systems. But blood-brain barrier is a lot more stuff gets through than we thought even a decade ago. So again, it's really interesting in terms of the physiology. Yeah. So I'm gonna make like just 2 more minutes. One is that this compression of the brain against the top makes it super difficult for us to figure out how much of this is squish and how much of this is neuroplasticity, because that's right where the lower limb somatosensory and motor control areas are represented, is right up here at the top and in between where the dura is getting pressed against these structures. So there's a lot of debate in this field right now. Is there neuroplasticity? Can we even measure it? Is it just squishing and moving? So I think that is a super interesting question and challenge. And then I, together with Manny Urquieta, you guys probably know, and my colleague at UF, Eric Du, we're setting up a human performance and neural testing lab at the Space Life Sciences Lab adjacent to Kennedy Space Center. So hopefully we'll get to do a lot more of this. And just want to make sure I acknowledge as well, of course, my incredible team members, grad students, and postdocs that really do all the hard work. So then I'll stop there. Oh yes, my helping hands in the shape of the brain. Yeah. Yeah. Okay. Great questions. I'm happy to answer more.
Pestered you a lot, sir.
What's that?
We pestered you a lot.
I know. I love it. I love it.
Well, I was wondering, would the opposite effect happened in diving people?
Hypergy.
But that's not pressure too.
Yeah,
Yeah.
it's also
Pressure.
it's also pressure. So
Well, that's what well, maybe that's what I was thinking of more than
more than like the floating
in my mind, when it gets inflamed because the pressure is.
yeah, so this is not necessarily inflammation per se, it's actual fluid shifts. So it's not the brain having an inflammatory response.
Yes, but it's not because, isn't that because the pressure has changed, so the
It's not clear that the pressure has changed, right? Like the
So the fluid moves
intracranial
different?
pressure. There's still debate about this, and we need better measurements in space, but the intracranial pressure does not seem to change. So it could be that, fluids are just distributed higher, which pushes the brain higher, which could be, pressure, or it could just be that it's floating higher. And then you're sort of blocking some of the fluid outflow. And so it's accumulating.
Maybe,
Yeah.
Estella, are you?
Yeah, I'm confusing them.
So
Yes.
it is
No,
true that they do
Mhm.
not run atmospheric pressure in the space station. It's lower pressure.
Yeah, I was,
So-
yeah.
And it is so more akin to being at high altitude. So the question is, I guess, maybe the question is, how do you separate effects due to being at low pressure?
Yeah. Yeah.
Yeah,
Barometric pressure versus the low gravity. Like, there could be effects that are related to pressure too.
yeah, for
Yes.
some reason I
Yeah.
was
And gas.
imagining.
Yeah, I know. And
Yeah.
like
Effect of-
CO2 and
All
yeah.
that stuff.
Yeah, right. So, right, it could be from anything, and we try to be careful about that in the papers. We say changes from pre to post spaceflight, right? So that's very agnostic to the mechanism
causal.
the causal mechanism. Yeah. And also a lot of our measures, our behavioral measures are in flight, but there's no MRI scanner up there, right? So we can't say the brain changes in flight. It changes from pre to post.
We need our MRI machine up there.
Yeah, or like better ultrasounds and yeah. But great questions. And I will say high altitude, like U2 pilots also have some
Yeah.
visible brain changes, not exactly this, but
Yeah,
also.
So about this funding matter, I thought
Yeah,
it was an interesting problem, but it's
Yeah.
true. So the people who buy their spaceflight ticket, some that go, for several days,
yes,
it's in the millions.
yes.
So
I know, let's cut out the
Wait.
middleman of the space company and
Well-
just have those people pick and fund the studies they want to be in. That's
I
my
mean-
view.
But I mean, I'm sure they could be bothered, like, to also fund their entertainment.
Right,
know?
right, exactly. I agree.
Although they probably see that, giving their time to the greater good, like they're contributing their flight ticket.
Yes, yeah, and I mean, it's tricky. There's actually one company out there that is charging scientists to recruit and consent their crews. And the others are not charging you, but they are not giving you any money.
Yes,
Yeah, so
I see.
it's, and I hate to admit it, we meaning scientists everywhere are snapping it up because
Data hungry.
you don't want to miss out. Yeah,
But also, when do you get by data,
I know.
Especially,
Yeah.
in that group of people. But, so then what is it? Like, should federal agencies step up and be funding these
So
studies?
Trish, the Translational Research Institute for Space Health, has an open call right now. They are the first
Okay.
to say, here are the topics we're interested in. Write your proposal for that. You will be connected by us with commercial flyers to run your study. And they're giving money to the investigators. It's a very small amount of money for what they're asking people to do, but that's kind of the way of the world, I guess, right now. So and NASA, I agree with this. NASA feels like they can't do it if it's not their crew on the private companies because they're
funded science,
but it's their mission to take care of their crew members, not to take care of private citizens. And there's a lot of discussion right now about whether the private citizens who have zero screening, I mean, they probably bought a note from their doctors saying, yes, you can fly, and are flying with all kinds of comorbidities, that what we learn about them might not be of use to NASA for protecting their astronauts.
So this work you
Right?
do,
It's so weird.
to be protecting, is that what it is? Because I figure it's like advancing our understanding of how
And
systems work.
that is part of buying down risks, right? We can't solve the problem if we don't know the problem exists. Yeah.
I see. So,
Yeah.
actually, okay, I see that.
Okay,
Awesome.
we're 15,
Yeah.
20 minutes over time. We'll
sorry.
finish the seminar here, but we'll continue the discussion.
Okay, cool.
Let's
Sounds
thank our
good.
speaker again.
Thank you.
Right.
Yeah.